So this is the North American medicinal leech. It has beautiful orange polka dots going down its back and black polka dots as well. Why? Scientifically, why? It's an interesting question. Is it to look pretty? Probably not. You agree it's a pretty leech, don't you? She's working on it over there. They look just like the black and reddish spots that you see on red-spotted salamanders, and or red-spotted newts. These newts are incredibly toxic to the things that eat them. And so by mimicking something that's incredibly poisonous, the leeches themselves, even though they're not poisonous, are actually protected by virtue of animals, like birds, for example, having a recognition structure where they don't eat things with red dots. And so they won't eat the leech. It's mimicry. Some of the most beautiful leeches that I work on, and I do understand this is an astrophysics class, we're going to get there, trust me, are marine leeches. These leeches feed only on sharks and rays. The one down here feeds only on sea turtles, and we don't know what those frilly things do. There aren't that many people who have been working on the biology or the physiology of leeches over time. I'm mostly working on the biodiversity of leeches and their evolutionary relationships, which requires that I actually go to remote places around the planet and go paddling off or hiking off to where there's water, where I can find them, rolling my pant legs up, wading into the water, and having the leeches attracted to me. So it's kind of an easy job, right? I don't have to go hiking all over. I just kind of have to wait there for the leeches to come. The wiggling is important because leeches will first sense you in the water mechanically. And that was one swimming towards me. You get out of the water periodically and see if you've got a leech attached to your leg, which I do here. This happens to be the giant Amazonian leech in French Guiana. Now, not all leeches are in fact aquatic. There are plenty of terrestrial leeches out there, and not all of them even feed on blood. This one that you're about to see is from Borneo.
Feeling up and down the worm's body, it searches for an end. Then, it starts to suck. The leech sucks its prey down like spaghetti, crushing the worm with its muscular throat.
Isn't that awesome? That's from the BBC Monsoon series that they did. You can get more of that if you like. Now, some of the work that I've done in the past actually concerns. I don't know if I've got the picture up here. Doesn't matter. There are leeches lots of leeches feed on various things, but that's helpful. But there are some leeches that are only specific to feeding on mammals. These are hippos. The only place that's got thin enough skin on a hippo for a hippo leech to feed from is its butt. And so this is actually hippo rectum. I'll give you the story about that in a minute. You can see the adult leech here. This is where the leech was feeding. And you see all these tiny little babies. So what happens is the parent leech, they'll lay their eggs under a rock and they'll fan those eggs until the young leeches hatch, and then they'll carry the young leeches to their first blood meal. This is parental care in leeches, things that not even all birds do. Don't let the smile on a hippo lull you into complacency. Hippos kill more people in Africa than all of the other animals, more than snakes, more than crocodiles. Probably not as much as malaria, but still, they kill tons and tons of people because behind that smile is a pair of very sharp incisors that if they get you in their mouth, they will snap you in half. Hippos are afraid of nothing. They're not even afraid of crocodiles. They're very ornery animals, and now you know why. They've got leeches up their butt. So we were actually in Africa trying to collect this for one of the stories I think I'm going to talk to you about. We're interested in the endosymbiotic bacteria in the leeches in various parts of the world, and so we had to get this particular leech. I did not reach up the butt of a hippo. Everything okay?
Oh, I'm just doing
Okay. I did not reach up the butt of a hippo. We were looking underneath the rocks to see if we could find them brooding their young. But I was going to go into water where there were hippos. And this guy with the rifle, I asked him, so like if a hippo chases me or gets me in its teeth or something, you're going to shoot the hippo, right? He says, do you see the caliber on this thing? That's not going to kill a hippo. But if a hippo gets you in its mouth, you may want me to shoot you. I think he was joking. We didn't have to test it. But like I said, not all leeches will feed on, are even interested. Most of the leeches around here are not interested in mammals whatsoever, and not interested in you. There are some places where that isn't true. So in the upper Amazon, in Peru, there were kids that were getting leeches up their noses. This is an endoscope going up a nose. It's going to go right back into the pharynx there. And you'll see there's a little leech hiding back there. And so kids swimming in this area were getting leeches up there, and it was actually quite painful. And when we got a couple of samples of it, we realized why it was painful. They have 8 very sharp teeth on their jaw inside their mouth. This is bothering you? Sorry. You can turn away. And we knew not only was it a new species, but it was also a new genus. And so I get to name these things, pick something out that makes some sort of sense. The word, Tyrannosaurus means terrible lizard, right? So Tyrannosaurus rex means terrible lizard king. Well, we decided to call this Tyrannodella rex, which means terrible leech king, mostly so that when we talk to the media, we could say that T-Rex is alive and well feeding on children in the Amazon. When I mentioned terrestrial leeches, the terrestrial blood feeders, you'll actually find them in places like Madagascar, Southeast Asia, Australia, Indonesia, the Philippines, places like that. We're doing something I think that's very interesting with these. We're going up, this is really getting bad now, isn't it? We're going off to places like Cambodia, places where the forests are actually being burned away for slash and burn agriculture, or where deforestation is happening for the purposes of creating rubber plantations and things like that. In order to protect a lot of these areas, what you need to do is figure out what lives there. Because if you're going to set up a national park, you need to be able to tell the authorities this is why you need to set it up. It's not just the trees, it's because of this animal. Maybe there are wild elephants still in this part of Cambodia. There are lots of other animals there too. What we're doing in terms of getting the leeches.
All right, so we noticed my pants were a little bit red, as you can see.
We wander into the forest, and again, the same sort of thing. We just use ourselves as bait. Normally we don't want them to feed on us. Then we can't use them for the purposes I'm going to describe. Couldn't find that one. They do tend to go north, and so I was like reaching up and seeing if I could find it. But fundamentally, what we're doing. I would love to think that the authorities in Cambodia are concerned about protecting and conserving the leech biodiversity in their country. No, that's not going to happen. But what we are doing is we're going into the guts of these leeches. They actually feed up to like 8 times their unfed body weight in blood. And we can find out what they last fed on using DNA sequencing technology. In fact, we can usually find out 3 or 4 of the last things that they fed on. And we can characterize the biodiversity in an area where there are terrestrial leeches by the leeches are going out and doing the finding for us, and then when the leeches come and try to find us, we gather them up in the hundreds, and we can figure out what is living in the forest. And I'll get to it in a second. The standard way to do this sort of thing is actually with camera traps. But just in the one place that we were in Cambodia, in terms of the DNA sequences that we got out of those leeches, these are the various animals that we found. Monkeys, bats, civets, small mammals, ground birds, frogs, rodents. Not one of these animals is going to trip a camera trap and get recorded by a camera trap in a forest because they're too small. If you set the settings so that you capture these guys in a camera trap, which records things going by, you're going to get every single leaf in gust of wind tripping the camera trap. One of the messages I want to give you guys, having seen the kind of science that I do, is to understand that, how many of you have heard of the scientific method, where you construct a hypothesis, and then you figure out what the null hypothesis is, and then you go and gather data, and if you can't reject the null hypothesis, then it didn't work or whatever. If you can reject the null hypothesis, so on and so on and so on. That's not how I do science. In fact, it's not the way most people do science. Most people do science the same way artists do art. You have a question or something you want to accomplish, and you get creative about it, and you think your way around it. You're passionate about it. With scientists, you go out and you make observations, and you gather them all together. Sometimes you're making observations randomly. You don't even know what the question is yet. And you write it down, and you figure out how you explain the patterns that you're seeing in nature, or in meteorites, let's say. And then you write it down and you submit it for publication and let the peers in your field evaluate it. Very few people actually operate according to the scientific method. I think in biomedical sciences, maybe more so. So this is a, sorry. I don't only work on leeches. I work on other worms as well. This is actually in Bermuda. I went to Bermuda in 2015, and I went there right around the full moon because there's the Bermuda fireworm, Odontocilus inopla, comes out on the third night after the full moon at 55 minutes after sunset, just with that precision, and bioluminesces in the water. That looks like this. See it? That's a female. She's going to swim in circles, and coming from over here, you're going to see a male, like a comet. And no, that's not an astrophysics reference. Like a comet. And when they get together like that, they dump their eggs and their sperm in the water, and this is how they accomplish fertilization. All of this solves a problem. A small, tiny worm in a big, vast ocean. How do you solve the problem of reproduction? One is if you're lighting up, you can find each other. And the males have really, really big eyes. And so that solves the problem of being in the same place. Doing it in synchrony with the phases of the full moon, which a lot of invertebrates, even coral reefs do this, solves the problem of time. So you have to be in the right place at the right time in order to get reproduction done. And that's how these guys do it. So we brought all the worms that I gathered back to the museum. We have state-of-the-art DNA sequencing labs at the museum. We sequenced, in fact, the RNA, which are the genes that are expressed at the time that we were pulling them out of the water and glowing, and we figured out which ones are actually associated with bioluminescence, and we even figured out the genes that are associated with that circulunar periodicity. You write it all up, you submit it for publication, it's reviewed by your peers, and this was published in January of this year. When you're looking, how many of you have gone to Google Scholar? Okay, so when you're trying to do research on science, how many of you have gone to Google? Well, come on, right? So this is searching vaping deaths, just because it's topical. And this is a bunch of stuff that you'll find when you search vaping deaths in Google. It might be a news story, it might be someone's personal story, it might be someone's crackpot opinion about what's going on, and so on and so on. Google Scholar is where you will find information that is done by scientists in controlled scientific ways, submitted for peer review to international or national journals. This is where the scientific literature is. This is where you want to look for the stuff that's actually out there in the scientific community. And here's a good example. Here's where we get into the astrophysics. So there was a lot of stories a couple of months ago. It was in August about tardigrades maybe living on the moon. It says, there live science, there are thousands of tardigrades on the moon, now what? Science alert, there could be a bunch of tardigrades alive on the moon right now, so on and so on. I got asked to comment on this. Here's the story about what happened. The Bereshit probe that was sent by the state of Israel was supposed to land on the moon. And they had a scientific study on there that had a whole bunch of tardigrades. You guys know what tardigrades are? Little water bears, moss animals. They're very cool. And I'll explain why in a minute. It crash landed on the moon. It didn't actually land properly. This is the spaceship that they sent. Oh no, sorry. This is actually the reason why they were doing it was years ago the European Space Agency actually put tardigrades into a contraption like this that opened up on the side of the Photon rocket, and for 12 days that orbited the Earth. And they were exposed to cosmic radiation. And cosmic radiation, do I actually have an example? Did I show cosmic radiation? I did, didn't I?
Yes.
You'll see it in a second. So there's a water bear, moss animal, less than 1 millimeter long. Why is this cool? Okay, so this is actually, is that going to play? There we go. Oh, do you want to hear the rocket going? When tardigrades lose their water, and they can lose up to 99% of their water, they go into a resting stage called the ton stage. That ton stage, you can actually boil them, you can leave them on a shelf for years, and you can just add water later and they come back to life. It's a terrific adaptation for an animal that often lives in mosses, where mosses can dry out until there's a rainfall, or in vernal pools in the southwest of the United States that maybe only get rain every 20 years or something like that. And there are animals that live there and can go into these resting stages. It's a state of suspended animation. Why does this make them so resistant to damage? Why is their DNA resistant to damage? Has to do exactly with the water. Ionizing radiation. This is actually at the Museum of Science in Boston. This is a cloud chamber. What you're seeing going through this ethanol, it's cold ethanol. I think it's cold ethanol. You see all those streaks? That's cosmic rays. And the reason you can see the cosmic rays right now, you've got cosmic rays passing through your body, and they're doing damage to you. Now, if you go up in a plane and you're closer to space, you're getting even more damage. So this is passing through you all the time. The reason you can see it is it's ionizing the liquid, which is to say it's actually converting it. The reason why DNA gets damaged from radiation is not because the radiation or the cosmic rays or X-rays or whatever are directly damaging your DNA. What they're doing is they're ionizing the water in your body and turning water molecules, instead of water, you get peroxide and you get superoxides. And these are the things that go and attack your DNA. So if you've got no water, then radiation can't harm you much. And tardigrades can get rid of 99% of the water. So what in fact happened with the thing that went to the moon was they put all of these tardigrades into a ton stage. They had no water. The idea was to expose them to radiation and then see how long see if they survive, just like had been done with the European Space Agency probe. So then the question becomes, with the crash landing of this thing, this is what actually happened to it, with the crash landing of the probe that went to the moon on the Parasite, it was almost down. It was almost landed, and then it didn't slow down enough, and it smashed into the moon. And in fact, there's sort of a scar left on the moon right now. You can see where that landed. Right, well, it probably landed here, and that's an ejectile thing. So the tardigrades they sent were not really in a living state, right? They're in a suspended animation state, so they're not going to be up on the moon crawling around. That's number one. Number two, when the European Space Agency did this, they had 3,000 tardigrades that they sent around the Earth for 12 days. Only 12% of them survived. It's not like all of them survived. And of the ones that did survive being rehydrated, all of them died over the course of 7 days. So what do you think the probability is of a high energy impact of a probe on the moon that had tardigrades in a suspended animation state that's now been exposed to radiation, cosmic radiation, without any protection for not 12 days but for 3 months, having any live tardigrades on the moon? Yeah, not very many. But this actually is something that should get you thinking about how a scientist like me thinks. Science is skepticism. Science is always questioning what people mean by what they say, and especially questioning your own findings. The mark of a scientist, the degree that somebody is a good scientist, often has to do with how much you're willing to disbelieve your own hype, your own ideas, your own data, and being able to specify under what circumstances would you admit you're wrong. If you can't do that, you're not doing science. You're doing something else, self-fulfilling prophecies or what have you. You must always be able to specify when you admit you're wrong, or you would admit you're wrong. I was a scientist very young. This is me when I was very young, probably just only about a month old or something. I grew up in Canada, in a small town. Spent a lot of time outdoors. This is just in our front lawn. My first science fair was actually, being Canadian, the science of making maple syrup. We went out. We had a ravine across the road from the house. Those are my mom's surgical nursing textbooks. I'll get to that in a second. So across the road, we had a bunch of woods. There were sugar maples there. There were black maples. There were silver maples. Well, it turns out that you can get sap for making maple syrup, not just from sugar maples, but silver maples I think work as well. Some not as good as the others. It's totally surprising to me that the maple syrup, the maple sap taps that you stick into a tree, that the hole is on the bottom, not on the top. I thought gravity would let it. Of course, what happens is sap comes from the ground up, and that's why the tap has a hole in the bottom so that it comes out. I think you get it. How much do you have to boil it down? Anybody else here make maple syrup? None of you. Okay, when you do it, if you do it, don't boil the water indoors. We took all the wallpaper off the walls in the house because you actually have to get rid of like 90% of the water before you get maple syrup. As I said, a lot of time outdoors, so it's not unusual that I become a biologist later in life. That's me in high school. I built my own kayak. A whole bunch of us did. We used to go on all these camping trips and canoeing trips in a place called Algonquin Park, about 4 hours north of Toronto. And even when you guys are seniors, right? So even when I was a senior or a junior in high school, once we had our driver's licenses and stuff, we would all pile into cars and just go on weekend or week-long canoe trips. That's my high school graduation day. And because I was interested in sciences, and I knew I was going to go into science at college, I went to the University of Toronto. I thought naturally, life sciences is what I'm interested in. I'm going to be either a doctor, or I'm going to be a dentist, or I'm going to be a veterinarian. I was set on becoming a physician, and went to the University of Toronto and worked my butt off. Now, in order to get into medicine, of course, you're going to have really good grades. How many of you want to go into medicine? Anybody? Okay, then to heck with your grades. No, I'm just kidding. You need good grades. You need good references. You have to have built up a CV in order to outcompete everybody. Well, freshman year in college, I got a C in calculus, and that wasn't going to work very well towards my overall GPA. And I was looking I was looking for ways to solve that. In the quad on campus, my friend Stephanie Keith, was studying for her finals when I was studying for my finals. I was kind of sweet on her. I'm not sure. To this day, I'm not sure if she knew that. I went over and sat down next to her to flirt, and she was actually reading this book. That she had it open to a page with some like really gross pathology or something. I'm like, what on earth? Forget about flirting. What on earth are you studying? What course are you in? She was a year ahead of me at the University of Toronto. I said, oh, it's a parasitology course. It's totally fascinating. And by the way, your biggest problem, because you want to get into medicine, your biggest problem is going to be that you have to take second year genetics like everybody else. This is a massive course that everybody in life sciences has to take in second year. It's impossible to know how you're doing because at the end of the year, the professor's just going to bell curve the grade so that, not everybody's going to get an A, some people are going to get Ds. And it's really, really hard, and it's boring, and it's terrible. But if you enroll in microbiology and immunology program as a double major, you can take second year genetics, or you can take this parasitology course. And there's like 30 people, and it's got a great prof, and it's fascinating stuff. And if you do well, you will do well. You'll actually know what your grade is, how you're performing through the year. So in order to avoid second year genetics, so that I could get a good grade and beef up my GPA, I enrolled in microbiology and immunology, not because I was interested in it, because this was affect a particular outcome, and I took the parasitology course. And there I was at the final exam period, and I'm in class, and the professor, Sherwin Desser, this guy, stern guy, he looks at me, he finishes a lecture, sort of wrapping up the course, and he says, Siddall. I said, yes, sir. It's like in front of the whole class. I want to see you in my office. And so terrified, I followed him up to his office, and he sat me down and closed the door, and he says, you've managed to do something that no one in the history of my teaching this course has ever done. And I'm thinking to myself, I fell asleep a couple of times in class, but He says, not only did you get 100%, not only did you get all the answers right on the lab exam, you got the bonus answers right as well. And I don't know if I'm supposed to give you 100% or 106%. I'm like, oh, that's good, right? I said, yeah, that's great. Listen, what I want you to do after the Christmas period is over, why don't you come back and join us in the lab, and we can find you some stuff to work on. We have these Ontario parasitology clubs and people from local universities. Is that warm stuff for me?
Yes.
Sorry, thank you so much. Where people who are doing research, graduate students and postdocs doing research, are sharing that with everybody. Why don't you come and join it? Looks like you got a thing for parasitology. And so I did. I joined the lab. And so these are the people that were working. Three graduate students working with Sherwin at the time. That's his laboratory assistant. That's my MCAT. I wrote the medical college admission test. And I did okay. I actually got an interview at McMaster University in the spring after that, which is to give you the next lesson. You guys are going off to college next year, right? I got lucky because he picked me out to come and talk to him. That doesn't happen. And nobody told me that if I really wanted to succeed in sciences, get to know your profs, the ones that are interesting, find out what research they're doing. Look at what they've written, what their graduate students are doing, and then go and talk to them. Get in their face. They've all got office hours. Take advantage of those office hours. And then he said to me, after a little while of hanging out in the lab, he says, look, there's this grant you can apply for from the Canadian government that will allow you to do research with me in the summertime, if that kind of turns you crank. Coming to spend a summer working on a scientific project at our research station up in Algonquin Park. Well, this was like the place I was going to when I was a kid. What a great opportunity. I'm going to go do research. And what a great line on my CV for applying to medical schools, right? I'm going to go work on blood parasites, malaria-like blood parasites of frogs. And so he asked me to work out the life cycle of something that hadn't worked out. And he says, it's probably not going to work out. People have tried and failed. It doesn't matter if you try and fail. You're an undergraduate. You're going to have a great summer. You'll learn a lot of techniques, blah, 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 blah. And so I did that. In fact, you can do this. If you're interested in sciences when you get into whatever college you get into, there's this program called Research Experiences for Undergraduates. It's funded by the National Science Foundation. There are research experience for undergraduate sites all around the country. I run the one at the American Museum of Natural History. We bring in 10 undergraduates every summer, and we put them on research projects. New science. What Sherwin did for me was he asked me to be a scientist. Go out and discover something that isn't yet known. And it was the first time anybody asked me to do that, as opposed to, here, learn all this science and give it back to me, and I'll give you a good grade if you got it right. Which isn't the same thing as being a scientist. So there I was, summer of 1988. This is Lake Saskatchewan. Down around here, there's a bog. And I was on the bog collecting frogs for this project. And I had a bag of squiggling frogs in one hand, and I had a dip net in the other, because that's how we were catching them. I was by myself, and standing on this stagnant bog that I could fall through at any moment. Beautiful sunny day like this, black flies buzzing around my head. And I had an epiphany. If I played my cards right, medicine that I've been working towards for like 10 years, I could get paid to do this for the rest of my life. And that's what changed. I didn't know that it was a possibility. I didn't know it was something that I could do. I didn't know it was something that I wanted to do until that moment. Never went to any more interviews. Withdrew my applications from various medical schools. Enrolled in a master's with Sherwin. So it turned out that the parasite, this is the parasite I was working on, and I worked out the life cycle that summer. People had tried for like 100 years and hadn't figured it out. It turned out it was leeches. Instead of mosquitoes that transmit malaria, for example, it was leeches that were transmitting the parasite from frog to frog. And I used tadpoles to do it. I ended up doing a master's with Sherwin on a similar malaria-like blood parasite of turtles. And again, it was leeches. A different group of leeches were transmitting the parasite from turtle to turtle. I was doing all the electron microscopy and cool stuff. That's me and a snapping turtle. And then I thought, well, we know that there are marine fishes that have these parasites too. And clearly the marine fishes are not being fed on by mosquitoes. It's probably leeches. And so I ended up doing my PhD on similar malaria-like parasites of marine fishes that ended up being transmitted by leeches. And accidentally, by doing all of that, I became the world's expert on leeches. Because I was studying the parasites and it kept being leeches. But it was totally by accident. Which is the next lesson. You must proceed by plan in an academic career, in college. Proceed with a plan, work hard. But you need to be prepared to succeed by accident. After that moment standing on the bog, my life has just become one tangent after the other, all because I was trying to avoid second-year genetics. And all of my research now is genetics of leeches. So I am responsible for a bunch of exhibitions at the museum. These are some of the traveling exhibitions: Power of Poison, Life at the Limits. I wrote a book about poison. It's a real page turner. It's kind of a bathroom book. You can open it up anywhere and start reading. There's no narrative to it. It's just really funky stories about people encountering parasites. The one that these images that you're seeing up here are from is the Picturing Science exhibit. And the idea that I had for that, I should probably drink this tea, not just look at it. You have to drink it for it to be good for you. Right. The idea was, I know from my years of being a scientist, that scientists look at their world with an aesthetic eye as well. And I went across all the divisions at the museum and I said, dig out your most beautiful pictures. I want to hang them in the halls of the museum as an art show. And show them off like it's an art show. And if people want to know what it is, they can go look at it and whatever. And maybe they'll learn something about why somebody was asking that question. But I know that when I'm putting my scientific papers together, I'm always looking not just for that image that is going to show whatever the scientific phenomenon is. I'm looking for the picture that looks the coolest, the one that is most appealing to the eye. Because we're all human, and scientists do it too. In some ways, science and art are similar. They're both trying to bridge the gap between humanity and society, let's say, and the world around us, and bring understanding to our lives and understanding of the context that we live in. So this was actually one of the images. You can see it over here. This is a star. Now we're getting into astrophysics, although I kind of did with the tardigrades, right? This is Alcor A. Alcor is part of a paired system. If you look up at the Milky Way, right up in here, if you look with the naked eye, it would look like one star. If you get a pair of binoculars or a telescope, you'll see that there's two stars side by side. There's Mizar and Alcor next to each other. Mizar is the bright one. Alcor is the one next to it. It's not a binary system that's spinning around each other. They're just next to each other in the sky. Here we go. I think that's a blow up. This is actually a picture of Alcor. Now, when you try to take a picture of a star, the question was, is there something around Alcor? Because they could see there was a bit of a wobble, and I'll explain the wobble later, of Alcor, suggesting that there is something else around Alcor. But when you take a picture of a star, you get a ton of light, and all that light interferes with the star is probably like some tiny little dot in here. And this is all the starlight that's coming at you. So how do you get rid of that starlight to see what's going on in here? And the answer, it turns out, is simple. Rebecca Oppenheimer, who took that picture, she's been doing a lot of this work, and I forget all the names of the projects, but basically it involves drilling a hole in the middle of the mirror. So the starlight's being collected by mirrors, right? By drilling a hole in the middle of the mirror, you're actually letting the central starlight through. Most of it, anyway. The deal is you've got to make a very, very precise hole in the mirror. If you just use like a hand drill or something, it's not going to work because it's going to create a lot of interference patterns. This actually, much of what you're seeing here I'll just bring it up this way. These are all interference patterns from the starlight. So even though there's a hole in the middle here, you're getting interference patterns from refracted and scattered light. Now, you would think that that's probably going to make it hard for you to see what's going on around the star, right? Same problem. What is an interference pattern? Anybody? Come on. You're embarrassing your teacher. An interference pattern.
Freshman year physics.
An interference pattern with light, right? Light of a particular wavelength interacts with light of a particular wavelength And where that wave is both high, you get a bright spot, and where they're both low, you get a dark spot. And you can see the bright and dark, these are interference patterns from overlapping waveforms, right? I would draw it on the board, but. Now, do you think that infrared wavelength, let's say like, I don't know, this long, is going to interfere the same way or the same patterns as you would get from ultraviolet wavelengths that are like this small? No, right? You're going to get different interference patterns for different size wavelengths. So why not take the starlight you've got left and actually look at one wavelength at a time? And that's what this is supposed to be showing. There we go. And you can see as you rocket through the wavelengths, longer and longer wavelengths, you're getting longer and longer interference patterns. That was also supposed to go again. But the stuff that doesn't move can't be due to interference patterns. And that spot right there that is in the same place regardless of the interference patterns is in fact a red dwarf that's spinning around Alcor A. This is Alcor A, this is Alcor B. And that was how they managed to take the image of it, which I think is pretty cool and ingenious. You guys know what this is?
Black hole.
It's a black hole. But you know what it is? It's actually an artist's rendition of the black hole. No one had ever seen a black hole. Black holes had been theorized for a very long time, and what was going on around a black hole had been conceptualized mathematically and from all kinds of information from radio waves from space. And so this artist came up with this as this is an artist's rendition of a black hole. There's a great role for artists in the interpretation of science. Until earlier this year, when Katie Bowman actually took a picture of a black hole. Do you guys remember seeing this? This was a big deal. Actually got a picture of a black hole from a distant galaxy. Did you guys see the look on her face when she actually, there was a lot of computation that went into putting this together, and she was waiting for it for a long time. And somebody managed to have a camera, or just their phone, and took a picture of her when it finally popped up on her screen. It's delicious. So when you when you have things spinning around distant stars, they're going to revolve around their central axis, right? The center of mass for both of them. So the star isn't going to necessarily just be staying in one spot spinning. If there's something big going around it, it's going to be pulled off of its center of mass, and it's the center of mass that's going to be the spinning point, right? So theoretically, you could look at a star, and if you see it wobbling, you know that there's something spinning around it, like a planet. It turns out you can't do this directly. If you try to do this directly, there's not enough wobble, and there's too much starlight, and you can't detect it spinning. But you can detect the effect of spinning. So here we've got a star and a planet going around, and when the planet's going around, when the star is moving towards you on this side, then the light waves are arriving more quickly, and you get a blue shift on the Doppler effect, right? When it's moving away from you, the relative speed is actually slower, and you get a red shift. So if you point a telescope at a star, and it's going blue, red, blue, red, blue, red, blue, red, you know that there's something making it wobble. You can even figure out how big that thing is. You can figure out how many of those things there are. The other way that we've been detecting planets is, in fact, by looking at dips in intensity. When a planet goes across, transits a star, you're going to lose a certain amount of intensity. And with a very precise telescope, you can figure out how much that is and how much is being included. But you can only do that with star systems that you're seeing edge on, right? If the star system's like this, you're not going to see that. It's only if you're going to get transit from the planet. So, in fact, the Doppler effect is the one that we mostly see. And what we know from those findings is that our solar system is actually really weird. Most of the ones we've found have got Jupiter-sized planets spinning in around the size of the orbit of something like Mercury. But it's also told us something about our solar system. We now believe, because of this and some other things, that Jupiter used to have a much closer to the sun orbit, and that something made it actually spin out of the inner orbits to the outer orbits of the solar system of the gas giants. This is just a sampling of various stellar systems that have already been figured out by astrophysicists, which I think is pretty cool. You can also start asking questions about, okay, well, It's a red dwarf and it's a blue star. A red dwarf actually has a very, very long life because it just sort of fizzles out. It never explodes. It doesn't have enough mass. Something like Alcor or Mizar, a blue supergiant, is going to turn into a type 2 supernova. Or maybe if the red dwarf, if Alcor is more like our sun, but the red dwarf gets sucked into it, you're going to increase the mass of that and change it to a different stellar classification. This is actually the spectrum of light from our sun. You don't get to see all of that. The only stuff you get to see is over here. This is all infrared and this is ultraviolet. And we usually work in, you don't really even get to see this because the lenses in your eye prevent the ultraviolet. It shouldn't surprise you that the principal molecule that's in your eyes, that's in bacteria, is in invertebrates for light detection on this planet, is something that absorbs maximally where our sun emits maximally, and that's definitely the case. So this is carotene and transretinol, and these are the molecules that are used for light reception across all of the animals on our planet, and that's the absorption spectrum relative to the emission from the sun. If you lived on a planet spinning around a red dwarf or a red giant, you wouldn't use these things. Our sunlight looks white to us because of this. Our sun would look blue to someone who evolved, or a group of organisms that evolved around a red dwarf, and they would be using something else. They wouldn't be using this. You know who this is, right? Galileo. Those are his early sketches of what he thought Saturn looked like, and he wrote it down and he shared it. That's what science is. Thanks to the changes that have happened in camera technology and imaging technology, we know that this is actually what Saturn looks like now, and it's beautiful. Much more beautiful than Galileo ever got to see. This guy's name is Daguerre. That's the original camera, and they were called daguerreotypes. One of the original pictures that he took with his camera and the first daguerreotype is a street scene from Paris. This actually, photography started in like the mid 1800s. George Eastman in Rochester, New York, came up with a different method, the method that was used up until digital cameras, and actually made taking pictures. He was the first person to really make taking pictures available to everybody like you and I by inventing the Kodak Brownie camera. Imagery, photography, has evolved quite a bit over time, to the point where now the thing that you've got in your pocket has way more power than an ability to take images than anybody thought was going to be possible even 20 or 30 years ago. And there are things you can do with that. If you want to take pictures of the night sky, there's an app that you can get, Nightcap. So the problem, obviously, with a camera like this is to take star trail pictures like this, or nighttime. This is actually a comet. There's a comet mode, or meteor mode, International Space Station mode, so you can take a picture of that as it goes by, or the trail of it is keeping the shutter open long enough and keeping it still. And so there's an app that allows you to do something like that. We talked about Saturn. This is actually Saturn in infrared. This is the red areas are actually the warmth coming from the inside of the planet. You can also look at Saturn, for example, in ultraviolet. These were pictures taken by the Cassini probe before it crashed into it. You can do this stuff with your smartphone now. This is actually an endoscope that you can attach. Actually, this works by Wi-Fi, so you can go and look at things inside of your ear or up your nose, go find a leech in the back of your throat or something like that. There are attachments you can get that will allow you to see in infrared. That's my son saying, hey, that's you. And pretty soon, this one's going to come out. This is an attachment for smartphones that lets you see in ultraviolet. You can't see in ultraviolet because of the lens in your eye. Your retina will see ultraviolet, but you have a lens that prevents that. Lots of animals, lots of things like bees and butterflies are seeing their world in ultraviolet. They don't see a sunflower like this. They'll see it like that because that's the ultraviolet signature. I've got a series of plants here. Regular light, ultraviolet, visible light, ultraviolet. And this is somebody applying sunscreen. Because of course sunscreen absorbs ultraviolet, right? It doesn't let the ultraviolet through And so you could use the thing to figure out whether or not you've applied sunscreen properly. We use ultraviolet imaging for other purposes in biology. It turns out that scorpions fluoresce under ultraviolet light. Lots of things fluoresce under ultraviolet light. We don't know why the scorpions do it. We do know that they're out at nighttime, so they're not exposed to ultraviolet light. What they're doing is they're fluorescing. So if you hit them with a black light, so that's just, that kind of looks green, right? And now it's fluorescing blue. Do you guys know what fluorescence is? When you put an incident light onto certain chemicals or minerals, somebody here has a red sapphire, pink sapphire, but check this out. What happens is the energy of the light is absorbed by the mineral. So watch this pink sapphire. Isn't that cool? Can you see that? It's absorbing the energy of the photons. That's causing electrons to jump to a higher orbital level. And then when they drop down to their resting state, they release that energy back as light. Now, it's not necessarily the same color of light that you put on it. It'll be something of a same or lesser energy because you're doing quantum state changes. So it's like quantum mechanics going on inside of a ring when I hit it with an ultraviolet light. This is a meteorite. And a similar phenomenon is happening here in terms of what you're seeing in these images. Instead of using the energy of light, though, an electron microprobe is firing high-energy electrons at particular spaces on these. These are chondrules on meteorites. The energy of the electrons that are released back, similarly to fluorescence, is characteristic of different elements. And so on this one, the red represents magnesium, the green represents calcium, and the blue represents aluminum. Which tells us a lot about this meteorite, because not all meteorites are the same. And what's really cool is that when you have those elements in common, the only time those got together in the formation of the solar system was near the very beginning, billions and billions of years ago. So that meteorite that landed on the Earth had been spinning around the solar system for billions of years before it got here. Oh, sorry, there it is again. As I said, we actually put this exhibition together to be an art show. I don't know how many of you are how many of you are art historian, have studied art and things like that, because it's clearly an homage to Andy Warhol doing it as art in series. Anyway, that was the story behind that. I just explained this meteorite as well. We can use other ways to image. We don't have to use photons. We don't have to use electrons, things with wavelength. What's going on here? These are this is the Grace Observatory that's running around the Earth, and it's 2 satellites, and they're communicating with each other by laser beams. What they're able to detect by changes in the speed of those laser beams. What are we done here? We're done at 1:30.
No, we're good. Sorry, we're just gonna take a couple panels to go get set up for physics, but you have time.
But when are we done?
5 minutes.
5 minutes. Okay. It's actually detecting differences in gravity of the Earth below. Different land masses, where there's more mass, you have more gravity. And so you can figure out with the gravitational attraction, minuscule changes in these highly precise lasers will tell you whether or not, what the amount of mass is below. And one of the most interesting things that's come out of this is changes in the mass of the Greenland ice sheet over the course of 15 years as detected by the Grace Observatory up spinning around the planet. And you can see where all the ice has been lost because of climate change. Wait, no, hang on. What did I just do? I pressed the wrong button. Okay. I want to leave you with a couple of thoughts about yourselves. Where am I here? There's some really cool things that people are doing now. This is, so Russians really like to have Dashcams. I don't know if you guys have gone on to Live Leak and looked at Russian Dashcams, all kinds of crazy things. But because of the way that we can do tomography these days, if you have a singular event, in this case, it's a meteor coming in over the top of Russia. You can see it there from a Dashcam. You can see it up here, over there, here, here. These are all different cameras in and around the same event, seeing it from different angles. If you know where the cameras are, then you can actually figure out where that line is in space. and you can follow it to where the meteorite impacted and dig the meteorite out, which is what they've been able to do because so many people have cameras. You can tell this is a meteorite. It's a terrible photo because of the scorching around the outside. So we cracked off the end here, and all that scorching is from coming through our atmosphere. So there are, if you're interested in getting involved in citizen science projects, there are a lot of opportunities to do that. This is smartphone astrophotography. Right now, you guys can go and be part of this. Take pictures of the night sky and share it with NASA. They're crowdsourcing information from citizen scientists like you. These are other opportunities. Silent Earth, taking recordings of where you are and uploading that to NASA so they can see what the noisescape looks like around the planet, or what the cloud cover is like. Or get involved with the Smithsonian's Global Telescope Network, where you can actually get engaged with looking at and analyzing data that's coming down from all of these telescopes from around the planet and do some citizen science stuff that way. There's in fact a lot of ways you can do this. There's a place called SciStarter that will allow you to go through and search for things that you happen to be interested in and see if you can get engaged, pardon me, as scientists on your own right now. And that's all I've got for you today. I hope that was
All right, guys, thank you so much. Red class, 7:45 tomorrow.